Well, hello everyone, and welcome to tonight's broadcast. My name is Eric Goldman, and I'm editor of Holistic Primary Care News for Health and Healing. And I want to thank you for all for attending this program tonight. The topic we've got on tap, CBD and cannabinoid therapy, is one of the hottest areas in all of medicine right now. It's been pretty hot for quite a number of years, particularly in the consumer space. We know that there's a lot of people who are using CBD and cannabis for a wide range of conditions. And over the last few years, I've seen major growth in the interest among practitioners in understanding what cannabis has to offer, what the various compounds are, and how to use it clinically. So we've got a really dynamite panel lined up this evening. I want to thank our sponsor tonight, Helix Institute for Health, for putting this together, and hopefully we'll be able to shed some light on some the interesting questions around what is CBD, how is it different from other cannabinoids, how do these things fit into clinical practice for managing and preventing and helping to ameliorate chronic conditions that you're seeing in your practices every day. Just to put things into perspective, we've got over 540 registrants for tonight's program, which, given that it's the middle of the summer and it's vacation time, I think it's phenomenal. It really reflects the fact that a lot of practitioners are interested in this. So if you're feeling like you're the only practitioner in town who's interested in what's going on in the CBD and cannabis space, you are definitely not alone. And in our 2018 practitioner survey, which we just completed in March, we had 469 respondents, and 56% of them believe that CBD and cannabis have medicinal value and should be more widely used, and 26% feel that it has value, but maybe it should only be by RX. And we know that in terms of prescription products for this, the FDA recently approved Epidiolex, which is the first cannabis-derived drug to be approved as a pharmaceutical in this country. So there's a lot of issues around that, but that is a very definite tipping point moment. I mean, I've been a medical journalist for over 30 years, covering all aspects of healthcare, and never thought I'd see the day that the FDA would actually approve a cannabis drug, but indeed they have. So we are definitely seeing a lot of action in this space. 24% of our respondents say that they are currently recommending or dispensing CBD in their practices, and 29% are recommending both CBD and cannabis. So this is definitely moving into the medical mainstream. 59% of our attendees say they want to learn about cannabis, and obviously you all out there tonight are among them. So we've got a dynamite panel, and again, I want to thank Helix Institute for Health and the folks at EnterHealth Botanicals for putting this together. Just a few matters of housekeeping. I know there's going to be a ton of questions tonight as we go through the program, but I'm going to ask you to use the little question box in your control panel there to type your questions, because obviously we can't take questions by audio, but we will try and answer as many questions as possible at the end of tonight's broadcast after we go through the formal didactic part. There is a handout. All the slides we've compiled into a single PDF handout, so you'll see in your control panel a little slot that says handout. Just open that and click on the title, and it should download the handout to your computer, and you can follow along, or you can look at that later. We are recording tonight's session, so a couple of days from now I'll send out a link so that you can review it, and all the people who haven't been able to tune in will be able to listen to it as well. And we are going to have a few poll questions throughout the program tonight, just to get a better sense of how you are currently engaging with cannabis and CBD, and what your clinical needs are, and how we can do a better job of serving you. So with that, let's start with the first poll question. I'm going to pull that up right now, and here we go. So the question is, how many do you currently dispense or recommend CBD and cannabis to your patients? I guess it's just, do you currently recommend CBD to your patients? So please select one of the following answers, and we will share that with everybody in a couple of seconds. Okay, so it looks like it is about just about half and half. 46% are saying yes, they're currently recommending or dispensing. 4% say they know, but they don't plan to. But obviously, you're out there and you want to learn about it. And then 48% say that you're considering it. So that's a really great start. And with that, I'd like to turn the program over to Steve St. Clair, who is the president and founder of Inner Health Botanicals and the Helix Institute for Health. So, Steve, let me see. I'm going to switch control over to you.
Great.
Okay, Steve, I can hear you.
Great. Thank you, Eric. This is Steve St. Clair. I'm, like Eric was saying, the co-founder of Helix Institute for Health. I'm kind of a serial entrepreneur, very dedicated to the health care providers in that industry. We have been involved in this area for many, many years. I just first up, I'm extremely grateful for everyone that's joining us tonight. This has been an overwhelming response. We had no idea what to expect, but this is just fabulous, we think. We're all here to learn more about CBDs and how they can potentially help your patients achieve optimum health. Thank you guys for joining us. Really, thank you. And also Eric and First Holistic Primary Care for getting this all together, the technology, the marketing, really appreciate it. So,
absolutely.
Yeah, and Helix Institute for Health is an educational entity that we put together to collaborate with speakers, people in the field, and we have some great speakers tonight. And the Helix Institute for Health will be offering webinars and classes in the future. What we hope is you can walk away with relevant information that will help you implement and or improve your CBD therapies in your practices. We also have a couple of other sponsors in the event. Helix Herbs, which is a brand, hemp CBD brand of Inner Health Botanicals. We have formulations that are very unique, using an ancient extraction technique that's been adapted to modern technology and FDA, compliant. Most of the bench chemists that we began this process with said that they wouldn't do anything. No, there wasn't going to be any value added, but they had to rethink their opinions once they saw the test results. This is a really innovative and potent technique. Now, Inner Health Botanicals is the parent company for the product side of this thing, and it was founded in 2005 and has been extracting over 100 single herbs. We have 30 different blends. The company that does the extraction is now called Inner Health, and it has gone by several other names over the past 23 years. Some friends of mine founded it 23 years ago, and I now have it. So Inner Health also offers a variety of nutritional supplements and organic functional foods, all manufactured in an FDA registered and inspected facility here in Colorado. The sponsors are offering a 15% discount on all orders placed by August 1st. So there's a lot of information. I just wanted to get that out of the way. Our first speaker with a lot of really great information is Luke Terry. He's a certified acupuncturist. He's a master of Oriental medicine. He's also an instructor of Oriental medicine. He's also has a practice, and he is an inventor. So I am going to turn it over to Luke, and I hope everyone really gets a lot out of this. And we would also love to hear feedback from you of what we don't cover in this and that you would like to see information in the future. Here you go, Luke.
Hey, everybody. My name is Luke Terry, as Steve introduced me, and I've been in practice as an herbalist and in functional medicine for about 15 years, and have been involved in health education for about 10, and have been involved in the cannabis industry in Colorado since the beginning of the legal hemp industry, which was in 2014. I got involved in 2015, and put some of my experience to work in the extraction field in Chinese medicine in some hemp extraction facilities, and became a consultant in the industry for the last 3 years. And I've been incredibly fascinated with what I've learned about the endocannabinoid system and using cannabinoid therapies. I've seen some incredible results in my clinical practice, and I teamed up with Steve as a co-founder of the Helix Institute for Health. So my job this evening is to provide you with some background information, to prepare the ground, as it were, to make the presentation a little more legible and to really create some context, hopefully for some deep learning here. So I've prepared some slides for you guys, and we'll jump into those now. Let's see, here we go. All right, so our main goals tonight will be to understand the general history of cannabinoids in historical perspective, and that will lead into a discussion of the endocannabinoid system as an overview. The endocannabinoid system is the reason why cannabinoids are actually valuable in our body, and it's a fascinating system. It's poorly understood, so I want to try to dispel some myths and shine some light on some of these fascinating interactions. The CB1 and CB2 receptors, among other receptors that are active, are G-coupled receptors. We'll explain what that means. We'll talk about the distribution of the receptors, talk about some of the other endocannabinoids and the related enzymes, the importance of terpenes, fascinating group of compounds that are present in many plants and insects as well, and they are a biological language of life. We'll talk a little bit about the cannabinoid pharmacokinetics, how they interact in the body, and then a brief discussion of the nutritional status, how that can interact with efficiency and effectiveness of both the endocannabinoids and exogenous cannabinoids that you might take into your body. And that will lead into Donna's presentation, and following Donna, we'll have Dr. Carlton. So jumping right in, historical context in Chinese medicine, the ancient physician known as Sheng Yang, he was called the divine husbandman. He wrote a Materia Medica in 2727 before the Common Era, so 2700 years before Christ. So we have over 4,800 years of using cannabis flowers medicinally in Chinese medicine. It's also been used for thousands of years in Islamic medicine and Greek medicine and in other Western traditions. It just so happens that in Chinese medicine, they had writing implements and writing paper, basically, bamboo sheaves and hemp paper that lasted longer than the things that they used to write on in other medical traditions. So we have a much longer context in Chinese medicine to understand how this flower was used. One of the earliest physicians to use it for pain relief was a man named Hua Tuo, and he is an iconic physician in Chinese medicine because he was both an acupuncturist and an herbalist. That's common today, but throughout the early era of Chinese medicine, physicians generally specialized. They were either acupuncturists or herbalists. Hua Tuo is a legendary physician, and he was a prolific writer, and he wrote a Materia Medica himself, and he described his famous recipe for what's called ma fei san, or boiling hemp powder. That was made from the flowers and seeds and has a described effect of being anesthetic and narcotic. It's a pretty famous formula. It was banned during the communist Chinese years, and a lot of that material was scrubbed from the Materia Medica. As in our own tradition, in the West, we used cannabis extensively medicinally in our pharmacies in the 1700s and the 1800s. In fact, it was a patent medicine, as listed in the US Pharmacopoeia until 1937. At that time, a tax stamp act was passed that made it very expensive to dispense these medicines, and that functionally ended the prescription use of cannabis for pain relief and for other things, even before cannabis was made illegal much later in the 1960s. So now we'll jump into the endocannabinoid system. This is an artistic representation of what the endocannabinoid system would look like. And it's representative as if it were a single cell. So we see a lot of different interactions here, and we don't have time to jump into these all today. We will review these extensively in our other course offerings that you can find out about at the end of the webinar. But you'll see on here a number of important interactions. We have the CB1 receptor, we have the CB2 receptor, we have the TVRP1 receptor. It's a transient voltage-gated receptor protein. It actually just appears when it's stimulated by the G receptor proteins in CB1 and CB2. We'll get into that a little bit more in a bit. We see interactions inside the cell. This is far more than we can get into today, but what I'd like to do is give you a few highlights that may help you understand the system clinically and help you prescribe cannabinoids effectively for your patients. So the first thing we want to know is, what is this system? And I've spent a lot of time trying to figure this out. And we hear quite a bit in the literature and in the marketing hype for a lot of hemp and cannabis companies that the endocannabinoid system helps to regulate homeostasis. Well, that's not very descriptive. Every part of metabolism that is regulated by negative feedback helps to maintain homeostasis. So that doesn't really help us. But having taken a deep dive into all this physiology and to understand what's happening in the system, I've come to believe that the endocannabinoid system is the endogenous reward and regeneration modulation system, because it affects systems across all different aspects of neurophysiology and immune and inflammatory regulation. It's quite a profound system. It seems to be something that is very, very central to our mammalian functionality, because it functions in all sorts of other mammals as well. So it's a very, very old evolutionary system, and it has a very high degree of activity and importance. And we know that if the system is disrupted, for example, by the French diet drug, which was a cannabinoid antagonist, it was a drug called rimonabant, it actually killed people by stopping this system by using antagonists. And they had to stop the clinical trial because 10 or 12 people died during the initial human trials of that drug. So let's get into a little bit more into this system and understand a bit more. The endocannabinoid system has a profound number of interactions with both the dopamine pathways and the serotonin pathways. So this is why I describe the system as an endogenous reward modulation system, because it has to do with everything to do with reward, pleasure, euphoria. It has to do with compulsions. It has to do with memory, mood processing, sleep, cognition. It's very, very profound, a lot of interactions. Again, more than we can go into in this brief period of time. So I've been trying to come up with a metaphor to describe what the endocannabinoid system does. So I've come up with the metaphor of an audio mixing board. And up here on the upper right hand, we see the inputs. The input system is something that can modulate across all of these different parameters, including serotonin, dopamine, GABA, glutamate, including many others, but these are the main four. So as the system modulates up and down, as our behavioral and contextual inputs modulate these sliders up and down, the endocannabinoid system kicks in to make these things more effective, to make them more powerful, to mitigate damage. And it's a overly simplistic model, I will admit. So if we really wanted to make this as complex as it would need to be, we would need a much more complicated mixing board, which I'll show you here in a moment. Our main two endocannabinoids that we're going to discuss today are going to be anandamide and 2-arachidonylglycerol, or 2-AG. And so we can see these then modulate across the board through various mechanisms. It modulates serotonin, dopamine, GABA, glutamate, and a host of other receptors. So as I said, if we wanted a truly inclusive model, we'd probably have a studio-sized mixing board that had many more inputs and a lot more stuff going on. It's more complex than we need to get into today, but hopefully that metaphor may strike a chord with you. So let's now take a look at the cannabinoid receptor site structure. The cannabinoid receptor structure is a protein that sits across the cell membrane. On the top, we have the extracellular side. On the bottom, we have the cytoplasmic side. It actually has two different binding sites. There's a main binding site, which we call the orthosteric binding site, and then we have a secondary binding site called the allosteric binding site. Both of these binding sites are active, and they both create downstream effects in the cytoplasmic side of the cell, and those effects are mediated through the guanine chain. We see guanine, triphosphate and diphosphate, similar to the ATP molecule chain that we're familiar with in the mitochondrial energy generation cycle. That's really how these molecules are, the direct internal cellular effect of these molecules. That's how it's taking place. And we've got a little animation here that should play here and show us how that works. So on the top, we have the ligand. That would be your cannabinoid, both endogenous cannabinoid or exogenous cannabinoid, fires the receptor. we get the G-coupled action with the adenylate cyclase, and that connects to guanine, and then we see the internal cell action there. That internal cell action can trigger all sorts of different mechanisms inside the cell that we'll explore later in further courses, but it can influence gene expression and inflammation and many other aspects of cellular physiology. So this is a really, really profound set of interactions in the body. As an herbalist that has studied thousands of different medicinal plants, 400 or 500 of them in great detail, I do believe that cannabis and cannabinoids are the most potent plant molecule that we have on a milligram basis that's widely used. I mean, there are some plants that are more powerful, but there are none that are this powerful and that have this high level of safety, which Dr. Carlton will get into the safety profile in his section. So let's talk about the endocannabinoids here. The main endocannabinoid is anandamide. Its name means bliss. It was identified by Raphael McCollum in 1992. The key fact here is that it's synthesized postsynaptically. That's a very, very important thing because it's not being centrally distributed. It is being synthesized on demand in the nervous system when conditions are optimal. The primary condition that you may relate to, that you'll know about when anandamide is synthesized, is the runner's high. So for many years, we believed that the runner's high was due to endorphins, but endorphins do not cross the blood-brain barrier. They do give us a nice body high, but the actual experience of being energized and being happy and blissful after exercise is in fact part of the endocannabinoid system. Anandamide is degraded by fatty acid hydrolase, and it's also THC's endogenous analog. So THC is fitting into the receptor site that is normally occupied by anandamide. So we see here on the left we have anandamide. Its molecular structure is very, very close to THC. So they're similar chemically, but their pharmacokinetics are quite different, which we'll get into here in a bit. The second major endocannabinoid is 2-arachidonylglycerol, or 2-AG. It's also discovered by Raphael McCollum in 1995. It is 100 to 1,000 times more abundant than anandamide. This substance is also a fatty acid molecule. It is also synthesized postsynaptically. It's an agonist at the CB1 receptor and a ligand at CB2 receptor, which means that it's going to have functions that are similar to both THC and CBD, or rather the reverse. We would say that their Venn diagrams overlap, but they're not identical. The interesting thing about this molecule, it is produced in response to glutamate stimulation and the resulting calcium cascade. And we know that the glutamate cascade is a very important part of normal nervous system physiology. It's the most common excitatory neurotransmitter in the brain. So every time you have a thought, every time you think about anything, or anytime you bring in sensory information, we are having a glutamate cascade. Glutamate is toxic. That's why MSG causes problems for some people because of glutamate toxicity and that resulting calcium cascade. And so 2-arachidonylglycerol's job is to help clean up the system and regenerate after the glutamate and calcium cascade from normal nervous physiology. It's very important in sleep, which is when we regenerate from all that calcium use. It degrades via MAGL enzyme into COX-2. So even its byproducts are anti-inflammatory. As you'll see, it's a very important part of the normal anti-inflammatory process of a healthy functioning nervous system. And when someone has a insufficiency in the production of 2-arachidonylglycerol, CBD functions just fine as an alternative. So this is an animation that shows a calcium cascade. So we're seeing nervous system tissue here, and there in the center you'll see a nerve cell firing, and the nerve propagation spreads throughout surrounding nervous tissue, and as it does so, we see that surrounding calcium cascade. As the screen is turning green there, we should also see in healthy individuals a cascade of 2-arachidonylglycerol. If that doesn't happen, the patient will not have proper nervous system regeneration, their sleep will suffer, they will suffer all sorts of different neurological conditions, and this is unfortunately very common. Distribution of cannabinoid receptors. We see the CB1 receptors are primarily found in the brain and nervous system, and the CB2 receptors are primarily found in the immune system and associated structures. However, the CB1 receptors can develop in other tissues, shown in 1996 that CB1 receptors can manifest in tissues as needed in conditions when it's called for in the nervous system. These receptors can also trigger other receptors to manifest, including that TVRP, the transient receptor potentiation cation channel. This is a very important receptor. The TVRP receptor is very involved in pain control. In fact, it's the major target of capsaicin. So it's actually maybe more potent than capsaicin in triggering this receptor, and it explains why endocannabinoids are important in pain modulation. Oh, skipped ahead too far here. Next, we'll talk about terpenes. Terpenes are an interspecies language of chemical messengers. Terpenes are produced in plants, in insects, in fungi. We have receptors for terpenes in almost all species, including mammals. They are incredibly easily volatilized, so they're easily airborne, and they allow species to speak to each other. Here in Colorado, there has been some fantastic research about communication in the soil between fungi and pine trees. It turns out that that communication happens via terpenes. Even in the animal kingdom, we have our wonderful herb catnip, and that produces a chemical compound that fits key in a lock with the feline reproductive hormone, which explains why cats go crazy when they smell catnip. They're actually being sexually stimulated. That is a terpene interaction in a mammalian system. The interesting thing about terpenes as it relates to cannabinoids is that they strongly modulate the CB receptor site, particularly CB2, via allosteric binding sites. So this means that the molecules that are in whole plant cannabinoid extracts that have high amounts of terpenes are more effective because they make the receptor sites more sensitive to the cannabinoids. And again, we'll dig more into that in some of the other courses, but we know that the whole plant extracts are much more effective than isolates. Donna's going to talk a lot more about that. Finally, we'll talk a little bit about the pharmacokinetics of exogenous cannabinoids. There's a bit of a mismatch here. Our endocannabinoids are synthesized postsynaptically, so they can be produced in small bursts very rapidly when needed in healthy individuals. When we take in cannabinoids from the outside, we're dependent upon bloodstream administration, and so we see a peak depending on whether it's orally consumed or inhaled or taken sublingually as to how rapid that peak develops and declines. In inhalation, we see that peak happens in 6 to 10 minutes for a total experience of 2 to 4 hours. That's whether we're inhaling CBD or THC or any other cannabinoid. Whereas orally, we only absorb about 6%, but the peak is 2 to 6 hours in, and it can take 10 to 12 hours. So for THC analogs, if we're trying to stimulate the anandamide system or mimic anandamide with THC, neither of these routes are very effective. But for CBD, it's actually quite effective, and people can benefit from really any manifestation or any administration of CBD because the peak is so much more gentle and because CBD doesn't have those pharmacological effects that impair people like THC does at a certain dose. You see there below on the left, anandamide's half-life is about 30 minutes. 2-AG has an incredibly short half-life, only 100 to 300 seconds, but it's constantly happening. As I said earlier, 2-AG is 100 to 1,000 times more prevalent. So we can really visualize the 2-AG system as just a constant flush of new 2-AG every time we use the glutamate system. So we can be safe in dosing CBD to replace 2-AG orally with a 12-hour dosage of administration. It's a nice mimic effect. Finally, to transition into Donna's, I'll talk a little bit about cannabinoids and nutritional status. Interesting things is that 2-AG is synthesized from arachidonic acid. As the name implies, it's 2-arachidonoglycerol. It's an omega-6 polyunsaturated fat. It is an important substance in that it requires omega-3 fats to potentiate some effects. Sorry, I'm getting a little ahead on my slides here. My effects are a little funny here. There was some research that was done in India a couple of years ago that is very clearly demonstrates that people that have defects in their omega-3 and or omega-6 pathways, either because of their genetics or because of their nutrition, they may see varying levels of chaos in their endocannabinoid system. So checking on a patient's nutrition and looking at their fatty acid status is an important part of assessing the endocannabinoid system. So with that whirlwind tour, I'm going to turn the slideshow over to Donna, and she's going to talk about some fascinating aspects of endocannabinoids. So Donna, you ready to join us?
Hello, can you hear me now? Sorry about that. My name is Donna Kuntzler. I'm a neuroscientist and a biological and chemical engineer. I really come out of the stem cell research field. I've been working in cannabis for several years now, and you'll probably notice that my engineering degree, when you see how dry my material is, relative to Luke's really exciting stuff. But anyways, the present culture of cannabis-based medicines has created a situation where the therapeutic effects, while evidently present, are being hotly experimented with within the scientific community. Initially treated with the same medical and scientific philosophy as a pharmaceutical, only a single or a few active ingredients, THC and CBD, were singled out to fill these roles. Much initial work was done on THC, which was thought to be the primary therapeutic constituent of cannabis. Later on, though, the non-psychoactive CBD cannabinoid or cannabidiol was identified as a possible candidate for the myriad of effects demonstrated by medical cannabis. Many studies that focused on these single cannabinoid isolates demonstrated minimal efficacy or sometimes no statistical significance whatsoever. And this is especially noticeable when you compare it to historically used wider spectrum extracts and whole plant extracts. This, along with a greater body of work I'll try to do justice in presenting, indicates that there's a lot more going on in these naturally occurring plant medicines than can be distilled down into a single miracle ingredient. Effectively, for now at least, scientists are dealing with a black box. The presence of these cannabinoids, as far as research is presently demonstrating, is necessary for the myriad of therapeutic effects currently attributed to cannabis, but they alone cannot account for all of the medicinal qualities. Cannabis extracts vary widely, and when you compare these, when you compare their effects, it's really necessary to understand whether or not they are a single cannabinoid isolate or they are what is considered a whole plant extract or full spectrum extract. Presently, it's not clearly labeled on the bottle. Even if you go to a website, maybe the supporting materials won't necessarily tell you whether or not it's an isolate or a whole plant extract or a whole cannabinoid extract. And even when they are termed full spectrum or whole plant, the diversity of the constituents and their concentration therein can vary greatly, depending on the type of extraction or extractions that the extract is derived from. Additionally, what solvent your extractors used, if you are prescribing this to a patient, should really be identified. If it is still present, and in the final product, anything that is present, that is liquid at room temperature will still be, just because of the laws of fugacity, will be present in the final extract. So you really want to make sure, like some extractors nowadays, you see them using isopropyl. Some of them you'll see using fat, but they heat it up to 200 degrees centigrade. I mean, and just denaturate. You really want to know what it is and what form that cannabinoid extract is really coming to you in. Anyways, the conditions of the extractions can also greatly affect the degree of decarboxylation, degradation, denaturation that the resulting extract constituent is subjected to. So, While THC and CBD isolates alone are obtaining like a lot of media and possibly hype, there's a large amount of reputable, peer-reviewed research being conducted that is beginning to show their real promise. Importantly, these cannabinoids, THC and CBD, show an improved efficacy when combined. These effects include anti-carcinogenic, antiemetic, and analgesic, as well as many others that have been multiple studies have reinforced the belief that this is actually a real phenomenon we are seeing. Additionally, CBD has been shown to mitigate some of what are considered to be negative effects of THC, including intoxication, increased heart rate, and sedation. This has been exemplified by Rousseau and Guy Jeffrey in their publication, but also Marceau shows the same effects here. It should be noted that the specific history of cannabis, particularly the outlawing of plant use for cultivation, has had a marked effect on the particular composition of these two cannabinoids. For decades, many of the people growing these were doing so illegally, and furthermore, a large majority were growing it to be used as a recreational intoxicant. As more money and interest in cannabis for this purpose increased, so did the drive for cultivators to select and hybridize plants to increase this potency of THC specifically. This resulted in a genetic drift in the population over time, which really favored higher and higher THC content. Both THC and CBD come from the same precursor, and their genetic basically, their genes determine how many copies they have of each enzyme, determine whether or not they produce more CBD and more THC. So in general, because you have a limited amount of the precursor, if you selectively hybridize or selectively breed for high THC, you're actually reducing your CBD amount. There's a lot of people who think we might have almost bred it out of existence for a little while there, but obviously that didn't happen. Of course, since this discovery of cannabidiol and the initial swing in opinion that it's now the primary active ingredient responsible for cannabis's therapeutic effects, the genetic pool of cannabis is expanding again, and cultivators are really now selecting for plants that do produce higher amounts of CBD. So here we see a figure from Marceau's and et al.'s study. Cannabidiol enhances the inhibitory effects of delta-9 tetrahydrocannabinol on human glioblastoma cell proliferation survival. This is published in Molecular Cancer Therapeutics, and glioblastoma cells, as many of you probably know, are possibly considered the most aggressive cancer cells that originate in the brain. As you can see here, the only statistically significant effect inhibiting these human cell proliferation is a condition that includes both therapeutic doses of THC and therapeutic doses of CBD. This trend has also been observed in many other studies, including ones that examine aggressive breast cancer, such as McAllister et al.'s cannabidiol as a novel inhibitor. I don't want to go through all these. I apologize. You probably don't want to hear all of this many list, but most of these are in Molecular Cancer Therapeutics and the Journal of Pharmacological Experimental Therapies. If you are interested in any more of this information, we will be providing it on the Helix Institute for Health's website. We will be doing subsequent classes as well. Furthermore, even greater efficacy than what we see in a CBD isolate and THC isolate is demonstrated when we do a CBD and THC enriched extract combination, which means I should probably take a moment to clarify that these extracts, while the primary extraction process focuses on either CBD or THC and concentrates them selectively, other therapeutic constituents, including terpenes, alkaloids, fatty acids, even the mineral content, are also left in the final product at concentrations that are still bioactive. We see this trend that these CBD and THC enriched extracts show a much higher efficacy when used in conjunction, as well as when you just compare a CBD isolate versus a CBD enriched extract, or a THC isolate when compared to a THC enriched extract. Some of the examples from literature include, up here we see CBD-enriched extracts here show increased efficacy and synergistic effects on the contractility of human and rat bladders when compared to just the in this urology. Sorry, in this urology article, that when compared to CBD isolates alone. Down here, we also see that these enriched extracts have a synergistic effect on intracellular calcium levels in hippocampal neurons and glia. These effects are much greater when we're using an enriched extract as compared to even a THC and CBD isolate compared. So even if they blend the THC and the CBD together and there's none of the other alkaloids, terpenes, fatty acids present, we still see a statistically significant higher efficacy when we have all of the other alkaloids present. This really does indicate that some of these other phytotherapeutics, possibly the terpenes, there's a lot of evidence for terpenes, the alkaloids, fatty acids for sure, there's a lot of evidence for fatty acids, are really adding to the effects and creating a plant synergy. Additionally, this last one down here, cannabis extracts show significantly greater effects in muscle relaxation in a mouse model for MS than a THC isolate alone. And even further, the CBD isolate by itself actually shows no statistical significant inhibition of spasticity. Also not shown here, but Elizabeth Williamson in her phytomedicine article, which I'll give you all of the citations if you need it, just reach out to me afterwards. Really shows that MS, once again, there's just more backup information on decreasing spasticity, especially in MS patients when they use a whole plant extract as opposed to just an isolate. It should also be noted that Galilei et al. Supposes that these CBD-enriched extracts follow a correlative dose-response curve, which is really like what we want when we're trying to treat somebody. You know, you increase the dose, and it should increase the effect correlatively. In this article, Overcoming the Bell-Shaped Dose-Response Curve, Galilei demonstrates that the CBD-enriched extracts, which once again is one of those whole plant extracts that has all of the alkaloids, flavonoids, terpenes, really provide increasing anti-inflammatory and anti-anxiety responses with increasing dose. Here we sort of see just a general view of a bell-shaped curve. When you use an isolate alone, you sort of max out at a pretty low dosage. Here we'll go to, this gives you, it's not exactly as clearly delineated, but here you see we tend to top out, and even increasing dosages of the CBD alone will not increase the efficacy. Note also, these are statistically significant, this one as well. When we use a CBD whole plant extract, which is primarily CBD, but also has the endogenous terpenes and alkaloids, increasing dosage increases the efficacy, which is really what a practitioner or anybody really interested in treating somebody, they want to, if you want to increase the dosage, you want an increased effect. These isolates, at least in this study, sorry about that, I'm getting a phone call, I apologize. So this really begs the question, what else might be present in these enriched extracts that may be contributing to their synergistic therapeutic ability? A lot of researchers are delving into this right now. Some of the therapeutics possibly contributing to these phenomena may be terpenes. As cannabis science develops and evolves, more and more attention is being focused upon these effects that cannabis's inherent terpene content has on the patient. Much of what defines the difference between what was considered to be a sativa or an indica, if anybody knows the difference, were up until recently, they were actually, most scientists, including geneticists, believe them to be completely different species. They believe the cannabinoid content may contribute to this. What really actually determines what was considered a sativa or an indica, sort of the sleepy or the more upper strains of cannabis, was actually the terpene set inherent in the strain, which really means a lot of these effects we're seeing, these side effects, might be attributed to terpene, and there is a lot of research contributing to that belief. One of the roles terpenes, as Luke already pointed out, have been thought to play is as a messenger molecule that modify homeostatic systems within an organism and communicate between organisms. Cannabinoids have also been thought to fulfill a similar role in the modification of different chemical cascades within the human body. A lot of peer-reviewed research is also suggesting that the alkaloids that still exist in these extracts may contribute to the therapeutic effects. Here we see Camelli and Francesca Camelli et al. Talks about how terpenes and flavonoids, basically she gets a statistically significant increase in anti and nociceptive or pain, basically I guess pain blocking effects than when using CBD alone up here. And then also Ethan Russo shows that the terpenes that exist in cannabis also exist at a pharmacologically active level, and they also have a statistically significant increased effect when present in their extracts. These are some of the most abundant terpenes in cannabis, though it should be noted that the variety, ratios, and content of terpenes can range widely, especially as commercial cultivators continue to selectively breed plants for smell and flavor profiles, and some of them are even cultivating plants to increase or mitigate particular effects on the strains they are developing. Beta-caryophyllene, this top guy up here, is a sesquiterpene, also found in a large number of edible foods and spices, cloves, rosemary, cinnamon, and can make up anywhere between 4 and 40% of the essential oil profile in the cannabis plant. It has also been shown to bind to CB2 receptors and act as an agonist, which means that beta-caryophyllene is also a non-psychoactive cannabinoid. Additionally, additionally, alpha and beta pinene. This is alpha. I didn't bother to show the beta. It's essentially the same molecule. They're also very abundant terpenes. And alpha pinene has been shown by multiple articles and multiple peer-reviewed research articles, which once again, I will give you all of the citations if you would like and hit me up later for it. It has multiple articles have shown anti-inflammatory effects in induced inflammation models. Beta pinene has been shown to help inhibit endocarditis and staph infections. And then also multiple down here, limonene is really an exciting terpene in the pharmacological world. Many peer-reviewed studies have shown that this monoterpene has anti-carcinogenic effects. And literally this research goes back to the 80s. We have statistical significance in everything, including many, many studies on mammary carcinomas. So it's super exciting. Limonene has also been shown to have antipsychotic effects. And there's even ER doctors now who are using it as a, people who in Colorado, where a lot of people are taking edibles, they're using it as a antipsychotic for cannabinoid-induced psychosis in the ER. Traditionally held views on plant medicines tend to favor whole plant extracts, and now we're really beginning to see a lot of research that is backing this up, and not just the terpenes, which we previously talked about, and not just the fatty acids, which Luke really brought up, but also flavonoids. There's a lot of research beginning to really examine flavonoids and how they really increase the efficacy of CBD and other cannabinoids. So CBD has so many targets of action. I am really not doing it justice in my list right now. I would like to point out that this is not an exhaustive list. Large amounts of research are being conducted on CBD and hemp extracts at this time. And as more research accumulates on this subject, a more refined view of the degree to which these properties may be attributed to CBD, or also possibly its co-constituents, terpenes, alkaloids, fatty acids, and then their synergistic interactions may emerge. But right now, there are multiple articles showing anti-metastatic, anti-carcinogenic, anti-inflammatory, antiemetic, anxiolytic, antipsychotic, also a decrease in seizures. But CBD is really only part of the picture. If you do not decarboxylate CBD, which means you really keep it raw, really in the plant, there's only THC acid and CBD acid. Processors generally decarboxylate it generally through heat, but there are other methods to do so. A lot of some of what we have initially believed as, well, anyways, basically we're really, our understanding of what is, can be attributed to CBDA, the acid form, and what can be attributed to CBD is really emerging. Initially, a lot of people thought like all of the therapeutic effects from THC, were from THC. Then they're like, oh, CBD, we're starting to discover CBD. Well, now as we really, really analyze the data we're being presented with, some of these therapeutic effects are actually from the acid form, the decarboxylated form. And as this understanding progresses, a lot of researchers and cannabis extractors are beginning to consider the nature of heat in the process of their extraction. Previously, heat was always applied because the decarboxylated version of THC got you high. THC acid got you high, and that was just automatically done by any extractor. But now, as we're really starting to look for therapeutic and medicinal effects, decarboxylation is not necessarily completely necessary, or even partial decarboxylation to leave some of these acids intact is actually desirable in a lot of cases. So as the role of non-cannabinoid cannabis therapeutics grow, greater attention to heat application from decarboxylation should be also, attention should be paid to that as well. Some of these other target molecules we've talked about, terpenes and alkaloids, have heat labile moieties that can be denatured at temperatures commonly used in hot extraction. Additionally, while researching the specific area of cannabis constituent degradation created by various forms of processing is progressing, molecules with similar structures to cannabinoids and terpenes denature at high temperatures and also with high exposure to gas-liquid interfaces and pressure. A lot of this research will be delineated in the future, but I'm going to go ahead and turn it over to Dr. Richard Carlton. And I thank y'all for listening.
Great, thank you, Donna. While we're doing this transition, I was thinking this is a good time to do another poll question. So I'm gonna go ahead and show my screen. Hold on just 1 second, and I just got to switch presenter back to myself. And we're going to show the poll question. So if you do recommend CBD, for what conditions are you currently recommending it? And the choices are chronic pain, seizures, insomnia, and sleep problems, support and cancer care, or I don't currently recommend CBD. And we'll just give that a couple more seconds. Okay, so it looks like 64% of you are recommending it for chronic pain, 24% are recommending it for seizures, 49% are recommending it for insomnia and sleep problems, 29% are using it in cancer care support, and about 30% of you don't currently recommend CBD. So very interesting. We know that people out there in retail land are certainly experimenting with it for a very wide range of conditions, and it's going to be interesting to see over time as more clinical research goes into this field, what conditions are most, I would say, amenable to or remedied by CBD and perhaps other cannabinoids. So with that, let's switch the control over to Dr. Carlton. Apologize for this delay, just having trouble doing the switch here. Okay, Dr. Carlton, can you hear us?
Yes, I hear you.
Okay. Okay, I am not seeing you show up in the control panel to be able to switch the screens.
If one of you has my slideshow, you could put it up and show your screen with my slideshow, and I'll just tell you when to change slides.
Okay. I don't think I can do that on my screen. Luke, can you?
If you'd like, I can do that.
Okay.
Yeah.
All right, here we go. You're gonna have to switch it back over to me and just tell me when to
Okay.
change slides.
Okay.
Okay. Let me do that. This is the first time we've done a four-person transnational webinar. We're all in where there's 5 of us on this, and we're all in different states, in different time zones. So it is a little bit complex. Okay, Donna, you should have control now.
May I ask? Can you see Richard Carlton's first screen now?
I do not. Yes, I do.
Now? Okay,
yeah,
perfect.
Yeah, bingo.
Lovely.
Great. Thanks for that.
Okay,
Okay,
thank
Dr.
you.
Carlton, it's all yours.
Okay, so I'm an integrative psychiatrist. I really wasn't interested in cannabis at all. I had stopped using in my early medical school years, but I got interested again because my wife has advanced Parkinson's and it wasn't responding to the normal kinds of meds. And as she got worse and worse, I got more and more desperate. And I saw these videos of people with advanced Parkinson's smoking a couple of tokes from a joint, and all of a sudden their bradykinesia just cleared up. They walked smoothly across the room, got into a car and drove. Their tremors stopped. I said, holy moly, there's something really incredible there. And as I began to look into it, I felt really deprived because I saw it was available in California, available in Colorado, and it was insane that it wasn't available in New York. It actually became available in New Jersey before it became available in New York, because I was very active trying to get it approved in New York. I testified before the state Senate hearings. I met with state senators to lobby for the compassionate care bill. I was very active, and I was actually helping children in New Jersey to get the medical cannabis from dispensaries in New Jersey by writing a letter, because they require a psychiatrist to write a letter in New Jersey for a child under 18 to get medical cannabis from the dispensary. So I saw it working. I was really intensely frustrated I couldn't get it from my wife. So if you go to the next slide, Donna. And I guess people, okay. So this is one of the girls that I treated right at the very beginning. This is Jenna. She has lissencephaly, abnormal convolutions of the cortex, 3 brain surgeries, 30 different anticonvulsants, vagal nerve stimulation, still having 50 to 100 seizures per day. And most of the time she wears a helmet when she's out walking. And that's what life was like. She's 14, but she's in kindergarten. Hit it down again, and you'll see after she started a CBD-rich strain of cannabis from the dispensary. Look at the difference in quality of life. She's gone down from 50 to 100 seizures a day to 1 seizure every 8 days. An important part here of this is that the CBD-rich strain helped to get the seizures down from 50 to 100 a day to 1 every 8 days, but that 1 every 8 days was a cluster of seizures, and the CBD wasn't stopping the clusters. So the mother experimented and got a THC-rich strain from the dispensary, and that would stop the clusters. So sometimes you have to mix and match. There's a lot to learn, and you have to modify doses, you have to modify strains, you have to be flexible in this wonderful field. Please go to the next slide. And by the way, I have too many slides. I'm going to have to barrel through this. I just wanted to, knowing that I don't have time to present all these slides to show any depth, but I want you to know that these things are there. You can go in and look at them afterwards. This is not a patient of mine, but this boy has an optic glioma, 14 months old, treated by a cannabis physician in California. On the left, you see the size of that glioma. I guess it's the size of an apricot. And he was hospitalized getting radiation therapy and chemotherapy, and this damn tumor was growing despite the chemo and the radiation. So I sent him home to die with hospice. Godspeed. Well, the parents got him to Cannabis physician who had the CBD-rich strain put on his pacifier. Several times a day, they'd replenish this pacifier. They'd just suck away the pacifier. Look what happens over the next 16 months. That tumor just disappears. The child's alive and well, and he was destined to die. They sent him home to die. Next slide. So you're really seeing the power of these cannabinoids. So Luke was talking before about the CB1 and CB2 receptors. A really important way to understand this. CB1 is mostly on our organs, the solid organs, including the brain. CB2 is more on our immune system cells, T cells, B cells, monocytes. When you think about anti-inflammatory, if you have immune cells, white blood cells, coursing through the bloodstream, going through all the different organs, and they're inflamed, with all the cytokines being activated, tumor necrosis factor alpha, all the interleukins, if you can occupy the CB2 receptor, either with your own endocannabinoids like 2-AG that Donna was talking about, or with the phytocannabinoid, CBD, if you can occupy those CBD receptors, you quiet down the immune cells that are inflamed, and therefore the organs which are coursing through, inflammation quiets down. So you have less inflammatory bowel disease, less fibromyalgia in the muscles, less seizures in the brain. You're quieting down and dampening inflammation with this wonderful system. Next slide. All right, so this is talking about safety. Hit it again. I want to show on the right-hand side. Donna, there you go. So the colored orange and red, yellow, those colors, that's this CB1 receptors in the brain. It's just really plentiful, especially loaded in the limbic system, basal ganglia. But that red circle, that's circling the brainstem. Very, very few CB1 receptors in the brainstem. They're there in the vomit center, so that's why THC can have such a potent effect on reducing nausea and vomiting. But they're not there. CB1 receptors are not present in the parts of the brainstem that control respiratory rate and cardiac rate. So you cannot die of an overdose that's from cannabinoids, from the phytocannabinoids. You can't die from it. It can kill you for other mechanisms, which we're going to talk about, but you won't stop respiration, you won't stop heart rate like will happen with the opiates. Next slide. So my colleagues have talked already about THC and the other cannabinoids and the terpenes and sesquiterpenes. THC is the part of this picture that gets the most press, gets all the oxygen in the room. But the CBD, CBV, CBG, the terpenes, that's such an important part of the therapeutic efficacy of the plant that it's really underrated. And when I talk to people, college graduates, grad school graduates, about cannabidiol, they say, "Cannabis, what?" So it's really underrepresented in the population. People don't know about it. Next slide. Keep going. Oh, so the entourage effect. Oh, sorry, go back one. Go forward. Those all together are called the entourage effect, a very important concept. Now, as was mentioned already by my colleagues, the endocannabinoid neurotransmitters are fatty acids, and that compares to the amino acids that we know as dopamine, serotonin. Those are amino acids. It's a whole new world of fatty acid neurotransmitters. And they have, over on the right-hand side in the white row, you see this intercellular action, presynaptic CB receptors, which are G protein coupled receptors, postsynaptic glycine receptors, but there's also intracellular action on the endoplasmic reticulum, the Golgi apparatus, and other non-receptor sites of action. So this is a world of complexity and of therapeutic efficacy. It has to do with receptors and non-receptors. Okay, next. Okay, skip to the next one, Donna. There's an important concept here of the circuit breaker. And there's an article with that title. On the right, you see a presynaptic neuron. On the left is a postsynaptic neuron. So of course, transmission is going from right to left in this slide. 2-AG is being released from the postsynaptic receptor and is going retrograde back up to the presynaptic neuron. It's hitting the CB1 receptor. And that occupation of 2-AG or anandamide on the CB1 receptor will shut down the calcium influx and therefore dampen overactivity in the presynaptic neuron. So here's two very important and significant examples. Let's say you have overactive glutamate transmission. Presynaptic neurons and the glutamate neurons are firing excessively, causing seizures. Well, the body has a built-in mechanism to dampen that excess of firing of the glutamate neurons by releasing 2-AG or AEA, anandamide, postsynaptically, and it goes retrograde back to the presynaptic neuron and shuts it down. In a book I'm writing on this subject, I liken it to smoking a beehive. So you get the bees calm. Now, it was mentioned before, I think it was Luke that mentioned that the CB1 receptor is very important, and that rimonabant was developed as a CB1 antagonist. They use it in patients who are obese, eating excessively, because you know how THC increases appetite by stimulating the CB1 receptor. So the reason, if we have an antagonist to the CB1 receptor, you decrease appetite, they lose weight. They did. What killed them wasn't the drug itself, because they got suicidal. And they had to stop the study because too many patients were suicidal and some of them were actively successful in killing themselves. So cannabis expert I know said, who would want to live with it? The CB1 receptor. A CB1 knockout is a devastating condition in animal experiments. And you want your CB1 receptor happy, you want it occupied. Next slide. Numerous controlled studies. I'm not going to have time to go into those. Please go to the next one. But the controlled studies show efficacy of these phytocannabinoids in MS, ALS, dementia, Parkinson's, pain. Oh, I should mention pain. So you have those same glutamate receptor neurons are excessively firing in your pain syndromes. Those nociceptive neurons are most often glutamatergic. And so by the same way, that circuit breaker concept, you can quench or quiet down pain fibers by the phytocannabinoids or the endocannabinoids. So it's also used in cancers of various types, seizures, GERD, Crohn's and ulcerative colitis, diabetes, PMS, etc. Next slide. Sorry to be whipping through this so fast. In many of these conditions, the heavy lifting is really being done by the CBD component. You don't always need THC. And part of my job as a physician is to help my patients understand when THC is really needed and when it's not. If THC is not needed, then A, you don't have to bother with the expense of it. You can just use some good CBD preparations. And B, you don't have to deal with all the psychoactive effects of the THC. When do you need THC? Well, first of all, nausea and vomiting and cachexia of cancer chemotherapy, HIV/AIDS therapy. There, it's really important to get that THC in there. Spasticity of MS very clearly responds to THC, but as Donna explained, CBD can help to modulate that effect. And of course, excruciating pain. Important to know, one of the best pain medications in the world and of medical knowledge is cannabis. Okay, next slide. It's the next one after that. It's a very good anxiolytic. I'm not going to have time to go through the spec results and the data, but keep going. So just to give you some clinical feedback, my patient Teresa said, I want to thank you so much for recommending this CBD oil. It's been so helpful for my anxiety, and I'm so happy with it. This is a woman I could not find the right kind of benzodiazepine and other kind of herbs, and I'm a fairly good herbalist. I was using all kinds of things to help calm her anxiety. Nothing was working until I got her the CBD oil. Here's a text message from my patient Wendy, letting me know her response to CBD oil. Upon awaking, decreased negative thinking, decreased worrying, clear-headed, not fuzzy. A week later, she texted, generally noticing decreased signs of depression, doing better in a gradual way. I am pleased. Next slide. Psychosis. Keep going. All right. So anandamide is important in controlling psychosis. I'm not going to have time to go into the details here, but you have these slides. You can go back and look at them. basically what's happening is CBD is increasing the half-life of anandamide. And folks, do you remember that you have enzymes that break down monoamines, like your monoamine oxidase inhibitors, or monoamine oxidase, and you have inhibitors of monoamine oxidase? Same thing here. There's an enzyme that breaks down our endogenous cannabinoids that's called fatty acid amide hydrolase. If we inhibit that enzyme, then anandamide will last longer. Well, CBD inhibits the enzyme, so anandamide lasts longer. I think the 2-AG does. I'm not certain about that. So bottom line, cannabidiol, CBD, is showing very superior results in controlling psychosis. Go ahead, next slide. And it compares favorably to sulpiride, another well-known antipsychotic, with no adverse effects and very good treatment of the negative symptoms of schizophrenia. Those of you who are fellow psychiatrists will know that our antipsychotic medications will stop the hallucinations, stop the delusions, but their patients are left with this anhedonia, slow thinking, blah, just a miserable existence, the negative symptoms of schizophrenia. CBD helps with those negative symptoms. And the doctor says, these exciting findings should stimulate a great deal of research. Yes, please go ahead. Go ahead. Go ahead. Okay, CBD appears to be a partial D2 receptor antagonist. Okay, dementia, go to the next slide. It's helpful in dementia. It inhibits acetylcholinesterase better than Aricept. It prevents aggregation of beta amyloid. We're talking about THC now. And one of the interesting effects of THC, smoked cannabis, in treating demented patients in a nursing home is euphoria. So they start eating and putting on weight, which is really important. There's reduced agitation. And when this was presented at a conference, one of the attendees said, America is a euphorophobic society. You know, what's the matter with a little euphoria? You have somebody who's agitated, screaming, hitting the nurses, let me go home, take me home. They don't recognize their own wife or their own husband. And you give them a couple of tokes and they're happy and they're not agitated. It's just beautiful. Go ahead, next slide. Insomnia, we're not going to really talk about this now, keep going. It really helps insomnia. It's one of the main reasons I use CBD. But I want to mention something important now. I'm really a big fan of EnerHealth's CBD products, and I was amazed to find that it's not sedating. Their CBD is not sedating. When all the other CBD products on the market that I've experimented with for me and my patients are sedating. So I asked their master herbalist, Leslie, why is it that EnerHealth CBD is not sedating when it had pretty much the same terpene profile and cannabinoid profile, CBG, CBV, etc., CBD? And Leslie explained that process that Steve was alluding to before, that they use low heat, no pressure. Steve didn't really talk about the pressure, but almost everybody in the industry, CBD industry, is using supercritical carbon dioxide extraction. What does that mean? At high pressure and high temperature, carbon dioxide, the gas, turns into a liquid and can then pull out your cannabinoids from the plant, dissolve them in that liquid, and then you decrease the pressure back to normal and decrease the temperature, and you have your CBD pulled out. But you've denatured the product to a very great extent. And EnerHealth, what they're doing is not denaturing the product. It's an old-fashioned, 300-year-old method that ancient herbalists used to use to extract herbal ingredients from plants. And I can taste the difference. I not only feel the difference, I can taste the complexity. To me, the other CBD products on the market taste dead, whereas the EnerHealth products taste very alive. There's a great complexity to them. If you know wine and can do wine tastings, you'll understand what I'm saying. Now in pain, not going to have time to go into the data in any detail, but take a look at the next slide. And you'll see that in pain of HIV neuropathy, there was a very significant decrease in pain in a marijuana cigarette. It's a cannabis cigarette versus placebo. And the next slide will show you a table that you can go into. Can you hit it again? There you go. So look later, and if you're interested and you see the statistically significant difference in pain scores when using this THC preparation and as a mixed plant. Go ahead. And reviewer says it's not a panacea, but cannabis stands ready at the vanguard of precision pain medicine. Isn't that nice? No, I agree. I want to tell you, I have a friend, a guy I know in the marijuana industry, who's a rock drummer, punk drummer, a big tats all over his body. He helps provide cannabis for patients who have cancer, and he himself fell off a ladder at work. He showed me his MRI. He's got bony protrusion into the spinal canal, and when his pain hits him, it's lightning pain on the floor, writhing, that kind of lightning pain. 1 or 2 tokes, and in a minute he's up, and he doesn't have to take opiates. So we're going to go to the next slide, and I'm going to skip that, just really release pain. That's what this is saying. So, is cannabis a gateway drug? That's one of the big objections that you'll hear. You're going to hear from your colleagues, you're going to hear from your politicians. They'll say cannabis is a gateway drug to big, heavy drugs like narcotics. In my experience, and talking to a lot of people, it's an off-ramp. It's an exit drug from addiction. It helps them to get off of opiates. And in fact, in states like Colorado, where cannabis has been legalized both medicinally at first and then recreationally, there's about a 25% reduction in opiate use, opiate overdose in hospital emergency rooms, and 25% reduction in opiate deaths. So people are actually able to use cannabis to get off of their opiates. That's phenomenal. New York State just added this last week a new indication for cannabis from the dispensaries, and that is to help people with opiate use disorder. So there's increasing recognition that cannabis, and that includes CBD, the plant phytocannabinoids, can help us to get off of the opiates. Next slide. We're getting near to completion. CBD is legal by an act of Congress to sell and to ship in the States, as long as it's derived from hemp, not marijuana, and it must contain less than 0.3% THC by weight. So that's a legal consideration. Next slide. Ethical consideration. From my mind, it must be organically grown, and it must not use hexane or other industrial solvents. And beware, some of the dispensaries in California and elsewhere, they'll use hexane to pull out THC, and then they'll sell that hexane extracted grass into the dispensary, and that's what the people are buying. So who the hell wants hexane? Even if you're healthy, you don't want hexane in your lungs, especially if somebody's sick. Now, supercritical CO2 is acceptable as a method, but not preferred because, as I said, it's too sedating. And my strong preference, as I told you, is for low heat, no pressure. Think EnerHealth. Next slide. Safety, we mentioned this already. Go ahead. Okay, some problems. You have to know the metes and bounds. I had a pharmacology professor, every single lecture, he would smile at the end, this little knowing smile, and he'd say, if it's strong enough to help, it's strong enough to harm. So you must remember that. So we must not become too enamored, too enthralled with our cannabinoids. There's always going to be some kind of problem as well. THC is absolutely contraindicated in unstable heart disease. The reason is that THC increases the respiratory demand of the cardiac tissues. If you've ever done a little too much grass or eaten too much of the brownies, the edibles, your heart was pounding. And that pounding heart, if somebody has unstable angina, can actually lead to a heart attack. And people have had heart attacks and died because they did THC. So if you have a patient with unstable angina or other serious cardiac problems and they say, oh, I just want a little toke, no. You got to come down really hard on them and you specify, I'm not opposed to marijuana. I'm in favor of it. Give all your reasons why you're in favor of it, but tell them, for you, for your condition, no, it's absolutely contraindicated. Next slide, Donna. Problem 2, THC can induce anxiety and even frank psychosis. So therefore, it is absolutely contraindicated in patients with schizophrenia or bipolar patients. And over the years, I've had a number of bipolar patients that just decided to smoke a little toke or two, and all of a sudden, within a week, they've quit their jobs, they're hallucinating, They're free associating. They're very fascinated by coincidences. Is the person who's talking next door really talking to them? Their mind just goes bluey with what ifs, and they're home living with their parents, and it's just a withdrawal, basically a decompensation that THC clearly induces. But CBD is the antidote to that. You must keep that in mind. Okay, next slide. Problem 3, THC and even CBD can induce hyperemesis syndrome. Now, some deadheads, people who are too dogmatic, pro-cannabis will tell you, oh, that's just stuff that they make up to give it a bad name. No, it's real. People who smoke or ingest cannabis on a regular basis can get hyperemesis syndrome, vomiting and nausea. It's improved by heat, and it's especially improved from hot showers, so they live in the showers. So it's provoked by cannabis. It eases up with abstention and recurs on partaking again in the cannabis. And I have a patient, 11 years old, who's on a lot of CBD for his grand mal seizures. The neurologist has him on something like 20 milligrams per kilogram, pretty high dose. He started getting the hyperemesis syndrome. So here's a little clinical vignette. Last month I started seeing a new patient. She's 32 or so. She was partaking cannabis for all her teenage years and 20s, got pregnant, gained 70 pounds, had gestational diabetes. After the baby was born, resumed taking cannabis, smoking, 2 bowls a day, and started throwing up all the time. And I said to her, because she thought it was anxiety and she was taking benzodiazepines from her physician. I said, do you take a lot of showers? Oh yeah, I live in a hot shower. I said, you have hyperemesis syndrome. She had never heard of it. Most of us haven't. Now you know, you really have to watch out for it. You're going to find it. Go ahead. Next slide. Problem 4, herb-drug interactions. This is very important. So this is a take-home slide. THC and CBD can be powerful inducers or inhibitors of some of the liver P450 enzymes that metabolize our medications. Now, plenty of things alter these enzymes. For example, many prescription medications alter those enzymes. Grapefruit juice alters them. That's why they say, don't take grapefruit juice for certain meds, and St. John's wort. So it's not a unique thing to THC and CBD, but it has kind of spoiled my fun because I'm really going home and prescribing the phytocannabinoids, both CBD and THC. But I've learned, okay, we've got to really look at what meds they're taking. And if they're in serious meds for severe cardiac problems or cancer problems or seizures, I've got to take a long, hard look. And every day now, I'm taking a long, hard look. Let's go to the next slide. And we're getting towards done. So some article here in Pharmacy Times says that THC and CBD have a limited ability to inhibit the CYP, that's one of the P450, CYP enzymes. They say pending additional studies, patients taking THC should be counseled to avoid drugs that alter the activity of CYP3A4 and CYP2C9. Smoking more than a couple of joints weekly is likely to result in induction of CYP1A2. Next slide. So if you're interested, go to this later on your own. We're going to skip over just some medications of which there's interactions. Now I want to tell you about some cases where I decided I would not recommend THC and not even CBD. And you have to be discretionary in this. So a 15-year-old girl, one of these New Jersey kids, and I to make it easy, these kids with seizures, I don't want them to have to take the Holland Tunnel into New York City and have seizures in the car. Remember, these are kids with 10, 15 seizures a day. So I do it by Skype video. So the parents take the Skype video camera into the girl's room. It's a darkened room, and she's on all kinds of medications, and she has fibromyalgia on top of everything else. And I find out she's on a fentanyl patch. So I say to the mom and the aunt, you've got to sign an informed consent I'm going to mail you, which says that you will discuss with the pain specialist who's providing the fentanyl patch that this girl is going to go on THC from the dispensary because there could be interactions with the THC with fentanyl. Let's think it through what happens. If the THC increases the breakdown of fentanyl, then you'll have less of an effect. right? You're breaking down, you're catabolically degrading the drug. You have less effect. So she's going to need more drug and therefore run the risk of an overdose. Alternatively, if the THC or CBD is decreasing the activity of the enzyme, the CYP enzyme, whatever it is that degrades the fentanyl, then you have a higher dose of fentanyl because it's not being broken down, and you're going to have an overdose. So there's going to be an overdose, a real strong risk. And I said, I'm not going to write the letter to the state of New Jersey saying that I recommend this girl as being a good candidate for cannabis from the dispensaries unless you sign this letter. So I sent them the letter. They never signed it. I called the aunt and said, I haven't received the letter yet. Okay, I'll talk to the mom. They never signed me the signed release. I never wrote the letter. I'll take the risk if they'll sign the informed consent that I notify them. But if they won't sign it, to hell with them. Okay. Next case, 80-year-old friend. Sad to see him. Got type 2 diabetes and heart attacks and ventricular tachycardias, many trips to the emergency room. So he's on a lot of cardiac meds. And I really thought this CBD would help him in a lot of ways, but I thought about all his cardiac meds. And I said, Bob, it's too many interactions. I can't figure out how the CBD is going to interact with your meds that you need to stay alive. So no CBD. Next case, 35-year-old ex-Marine with something like undifferentiated schizophrenia. When I interviewed him, he talked like this in a whisper. I don't know if you can hear me, but this is how he talked. And I would say, could you repeat that? Wow. He just couldn't talk in a normal tone of voice. And he'd been hospitalized twice while he was still in the Marines. And I said, what were you hospitalized for? Well, I don't know. Very vague. So I said to him, I can't prescribe the THC for you, the cannabis from the dispensary. I'm just worried it's going to make you psychotic. And he said, well, I smoke it a lot and it doesn't make me psychotic. It makes me feel better. I said, well, I appreciate that, but I'm not going to take that risk. I think there's a strong chance it could really induce psychosis. Maybe somebody else will prescribe it for you, but I in good conscience can't do that. Finally, 35-year-old ex-con who called 2 weeks ago on methadone, PTSD. What's his PTSD from? A gunshot wound. He's been in the penitentiary, and so for fear of my own safety, I wasn't going to prescribe medical cannabis for him in the New York State system, but had to give him a sensible reason, and it's a true reason. I said, I don't have sufficient training in the management of opiates because he's on methadone. So I'm sorry, I can't do it. I don't know if you know this, but a number of the pain specialists who deal with patients who are getting opiates a lot of the time, they've been shot, in other ways attacked by their own patients or by the spouses of patients. If they declined to continue the opiates, they've been shot, and a lot of them are carrying guns now. So I won't prescribe patients who have a criminal background like this, just for my own safety. So next slide. We're getting near the end. Limitations of THC. Go ahead, next one. Managing the high. Taming THC is a wonderful phrase by Dr. Ethan Russo. Next. So the ways to counteract too much THC. CBD, that's your mainstay. That's what you go to. It competes with THC for the CB1 receptors, and it competes with FAAH, that enzyme, so that anandamide is not broken down as much. So you counteract the high. Fresh squeezed lemonade, because it has the pulp, is what you want. It's rich in terpenes. Limonene, and I was discussing this with Donna last week in a dress rehearsal for this conference. Donna pointed out to me what she said to you already, that the emergency rooms in Colorado for people who have taken too many medibles, those edibles, and they're psychotic, they use limonene. It's terrific. Pistachios, ostensibly, and PEA. This is a really important molecule for you to know about. Palmitoyl ethanolamide. Remember we talked about fatty acids are the precursors to these endogenous cannabinoids, endocannabinoids. So arachidonic acid, Luke talked about that. Now we're talking about palmitic acid, oleic acid. They're all precursors. So PEA is palmitoyl ethanolamide. It's a cousin of anandamide. Increases levels of AEA, anandamide, allowing AEA to compete with THC for the CB1 receptor. Reduces pain and is calming, but is not psychoactive. And of course, if you have somebody who's really high from the THC, you could prescribe a benzodiazepine. Next slide. Managing the addiction to THC. Go ahead, next slide. This is N-acetylcysteine is a very good way. This is the current issue of Current Psychiatry, lead-off article. Next slide. And I'm not going to read you the details, but basically their findings support NAC as a pharmacotherapy to complement psychosocial treatment for cannabis dependence in adolescents. Basically, N-acetylcysteine is anti-glutamatergic, anti-compulsive, and cannabis addiction, like many other addictions, it's an addiction. It involves the glutamatergic system. NAC helps many kinds of compulsions and addictions. Next slide. Dosage guidelines for THC. 5 milligrams is a pretty reasonable starting dose for somebody who's had prior experience with THC. If they're naive, a much safer starting dose would be 2 milligrams or even less, just microdosing. CBD, 5 milligrams is a reasonable starting point. Now, if you get the 250, the EnerHealth hemp concentrate, 250 milligrams per bottle, 1 dropper full is about 6 milligrams. So a dropper full of their product would give a very good reasonable starting point. Some people have to microdose with just a few drops. I was giving a talk last year to the American Psychiatric Association, New York City branch. A young woman physician came up to me before the talk and said, she's using CBD for her migraines. I said, terrific. And she says, the only thing that's worked, none of the meds worked. CBD works. I said, great, what's your dose? She says, 4 drops. I said, you mean 4 droppers full? No, 4 drops. And how could that be? But you hear about microdosing. And when she at one point cut it down to 2 drops, that wasn't enough. She had to go back up to 4. In refractory pediatric epilepsy, on the other hand, neurologists will take the dose up to, I think it's something like 10 milligrams per kilogram. So there you want to go to really high doses. Go ahead, next slide. Almost done. Vape, 1 minute to onset, 2 hours to offset. I don't recommend vaping. It's dangerous to inhale the antifreeze they put in it, propylene glycol, especially when the damn stuff is heated to 200 degrees Fahrenheit. We don't want that in our lungs. So I like the oral mucosal, the sublingual. You put it under the tongue, hold it there for 2 minutes, and then swallow it. 3 minutes onset, 3-hour offset. That's pretty close to what the vaping is, so it's very fast. I don't like the oral route because, as Donna was showing before, the liver converts delta 9 THC to delta 11. Now that is way more psychoactive. And so what could be a very sweet trip where music sounds nice and the art looks beautiful and wow, far out kind of experience, lovely, and you get a little bit hungry and you laugh at things, the delta 11 can be nightmarish and paranoid. And because it's a long time to offset, 8 to 12 hours, that's a nightmare. And people jump out windows. And one of the main things you have to prepare your patients for, if they're going to do oral, if they're going to tell them they're going to go to Colorado and go to those little stores and dispensaries, you tell them, if you take the oral, those little gummies, you just do 1, you don't take more than 1, and you just wait 2 hours or 3 hours, and you don't smoke a joint, you don't take another gummy, because people are impatient. They're used to getting a very rapid onset from smoking. They're not used to waiting 2 hours, so they're impatient. So they take a second dose of the oral and a third dose. And by the time it hits, they're jumping out windows. It actually has happened. So Colorado has a law which said that 10 milligrams is the maximum that they're allowed to put into a gummy. And even that's way too much if you're taking 2 or 3 of them. But here's an interesting idea at the bottom. You look at this little bullet point. Let's say you have a patient who needs continuous coverage. Let's say they have unrelenting cancer pain. So if they can tolerate the oral route, they don't get nightmarish anxiety and hallucinations from the oral route. So you can start them off with the sublingual, let's say the dropper full, and at the same time they can take the oral, the capsule, or the gummy. So by the time the sublingual wears off in 2 hours, now the oral dose is kicking in. So in that way, they're getting long-term coverage. 8 to 12 hours of coverage. Then they can do the same thing 12 hours later. Next slide. I'm not going to have time to go into this, but just one real quick thing. 3 members of the same family have Crohn's disease. Not my patient. he came to me for obsessive compulsive disorder, but he, his brother, and his mom all had Crohn's. They all were on 6-mercaptopurine. They all went to a dispensary, legal in New York, and got a high CBD, low THC strain of cannabis from the dispensary. All three of them got off of 6-MP, and they're completely cured of the Crohn's disease. No 6-MP, no symptoms. I have a neighbor with cerebral palsy, and it's helped him tremendously. It's just wonderful. And most of this is CBD. Next slide. Here's some educational information, and Steve is going to tell you in a minute about what Helix is doing, which we'll add to that. Next slide. Here's some books. I'm not going to have you read this now. And the next slide is my book that's pending. If you send me an email and just in the subject heading say book in capital letters, I'll put you on a mailing list for when the book is published and let you know. It's Medical Cannabis, the Promise, the Evidence, and Ways to Manage Its Limitations. Next slide. And that's it. Thank you so much for your patience, and sorry for taking a lot of your time there. Steve?
Thank you, Dr. Carlton. Yeah, as Dr. Carlton was alluding to, the process that we use is hundreds, if not thousands of years old. It's actually I think, and Donna thinks, it's really the only true whole plant extract, because we do 2 extractions in ethanol. One of them is an ethanol extraction. We use organic ethanol and organically grown hemp. And no, the heat never goes over 98 degrees, which gives us a better profile of terpenes and flavonoids and things like that. We also retain a lot of the A forms, which is optimum in my opinion. You need a combination of both. But then we do a second extraction where we're extracting the, as Donna calls it, rare earth minerals and the salts from the plant, and we reintroduce that back into the original extract. So we're using, I think the correct word, Donna, is endogenous material.
Yes.
And I think Donna has some research that's actually been done that shows that these, I guess you could call them hemp or CBD tinctures, are actually more effective with ethanol and salt.
Yeah, the highest clinical bioavailability found for CBD so far has been using a small amount of salts and ethanol to increase basically just penetration past the blood-brain barrier and also intramembrane.
So the process that we use is everything that's in the plant is in the bottle. And it actually, as Dr. Carlton says, it tastes like it's still alive. And my kind of working opinion right now is I think the plant thinks it's alive. It's in the bottle. It's just purified. Everything that was in the original plant is in the bottle, just purified. We're actually doing some studies to try and define exactly what's going on, because, like I said, most of the bench chemists said nothing would happen. But the chromatographs of this product, when you look at a before and an after chromatogram, radically different. Radically different. It's amazing, truly amazing what happens. The activity increases 10 to 50 fold. So, we've had one chemist come up and said, I think fractionalization is going on. There may be some esters being created because the rare earth minerals, there is everything that's in the plant, including we feel that there is a minute, like nanomolecule of sulfuric acid, which when exposed to ethanol creates a Fischer esterification. So there's a lot of things that we don't know, but we do know that it works like Dr. Carlton's saying. It works, and it works very well.
Great. Well, thank you, Steve, and thank you, Richard and Donna and Luke. A lot of information on a lot of different levels, from the molecular biology of what goes on in the brain and the endocannabinoid system, all the way up through how do we apply this practically in clinical settings. I imagine some of you do have questions. Please type those in, and we'll try and answer as many as we can before we hit the top of the hour. We do have a hard stop at the top of the hour. I have a couple of questions that came up as you were all talking. What accounts for the appetite stimulating effect that many people experience with cannabis, particularly whole plant or smoked cannabis? I don't know if a lot of people who use CBD alone are having that similar effect, but can you say one of you say something about that?
Yep, I can answer that, Eric. There's a particular pathway in the CBD reception system. It's not actually CB1 or CB2. It's a receptor called GRP55, and it has to do with appetite regulation and satiety. And it turns out that THC and CBD have opposite effects on that pathway. THC upregulates it, thereby upregulating appetite, and CBD downregulates it. So CBD is actually shown to be effective in a couple studies for weight loss.
Okay, I was going to ask about that. Does it have a place in weight management?
It does appear to. That's correct. And there's also an interesting effect as well on another pathway. That's the PPAR pathway. PPAR alpha and beta. That's the peroxisome proliferation and activation receptor. It's a generalized lipid receptor that's in all of our cells, but particularly dense in the nervous system and fat cells. And that system can be upregulated via the endocannabinoid stimulation as well.
Great. And I have one more sort of clinical indications related question. I believe it was Donna, in your slides, you said something kind of in passing about the possible role of cannabis and CBD in multiple sclerosis.
Yes.
Can you say any more about that? And what about other neurodegenerative diseases, Parkinson's, Alzheimer's, dementia, things like that? And, Dr. Carlton, please feel free to weigh in on this as well.
Well, I'll give you, can you hear me? I'm sorry. I'll give you a
Yes.
little bit. I mean, Dr. Carlton probably will have a lot more information. Many studies are actually showing that it helps with inflammation and pain, but also decreasing spasticity in multiple sclerosis. And then like Dr. Carlton brought up in dementia and Alzheimer's, it actually blocks the formation of beta amyloid proteins, plaques on the brain, which effectively is one of the main measures we use to sort of determine the progression of Alzheimer's. So blocking it also not only blocks that plaque formation, but it also seems to slow the progression of Alzheimer's on a sort of on a person to person, like, you know what I mean? Sorry, how their quality of life, and then also how their family members are interacting with them. But Dr. Carlton probably has more he could add to that.
Well, so it's really interesting. The holy grail in medicine is to find disease-modifying processes that will not just treat palliatively, like reducing pain, but actually slow down the progression of the disease. So in Alzheimer's, THC is a disease-modifying agent because it's stopping the aggregation of the beta amyloid, and also because it's increasing the levels of acetylcholine by inhibiting acetylcholinesterase. So you're actually slowing down the disease process. Interestingly, the same thing is also true in rheumatoid arthritis, because you're quenching that inflammation, not just in the joints, but in all the white blood cells going through the joints that are activating the joints, all the cytokines that they're pumping out, whether they're in the joints or not. They're all pumping out cytokines. So if you quench that inflammation, you're actually slowing down the disease process and the destruction of the joints in rheumatoid arthritis. Yeah, and just to finish, in Parkinson's, there's a lot of CB1 receptors in the basal ganglia. Remember that slide that I showed you of the PET, I think it was a PET scan, showing the receptors. So the net effect is a smoothing of the firing, a coordination of the firing. I know it's kind of vague, but what that shows us clinically is a decrease in bradykinesia and a decrease in tremor. and it's
Wow.
very noticeable. Fascinating aside, in different stages of Parkinson's, you have different levels of CB1 receptor expression. I think there's a lot of CB1 receptors expressed early in the disease and fewer CB1 receptors later in the disease. And that poses some interesting questions and challenges. Well, is it does the brain want more CB1 stimulation early in the disease, so it has more receptors? And does it want less CB1 stimulation later in the disease? It's interesting, but there's a lot to learn. But the bottom line is these cannabinoids can help in the movement in degenerative disorders.
Great. Well, I want to get to a couple of the questions that have been coming in from our listeners. Charlene is asking, by Crohn's patients being, quote, completely cured, do you mean that the patients no longer need to take the high CBD and low THC compounds as well? I mean, what counts as a cure? And also, related question, what concentrations of CBD are you using in the Crohn's context?
Okay, so that was not my patient. He was my OCD patient, but I didn't prescribe the cannabis for him or his family members. He had gotten a ratio with something like 4 parts CBD to 3 parts, 2.5 parts THC, 4 to 2.5. So it's CBD predominant from a dispensary. And it's a good question. I don't know if he continued to take it. I believe he did. But all I know for sure is that he was able to go off to 6-MP, as was his brother and his mother. And they have no symptoms.
Wow. Okay, we have a question. Let's see. Greg was asking earlier on the broadcast to get a little more clarity on what is the G-coupled factor again. That was earlier on when you were discussing the endocannabinoid system. So I think that was Luke.
Yeah, the G-coupled receptors are quite common. Many of our neurotransmitter receptors are G-coupled proteins, and it means that there's a guanidine triphosphate or guanidine diphosphate activity inside the cell. So when the ligand, like anandamide or 2-AG or THC or CBD, when it stimulates that receptor on the outside of the cell, it triggers the release of that guanidine triphosphate inside the cell, which then causes those internal actions and eventually reports the activity to the nucleus of the cell, where you get protein transcription and translation. So G stands for guanidine.
Okay.
And if you don't mind me adding, a lot of these G protein, they're slower acting. They tend to do more homeostatic changes, so they're not like quick acting. They make longer lasting changes. So their effects sometimes are nutritional, and sometimes they cause complex chemical cascades, but they're not fast moving. They create a different state within the patient.
Great. Thanks for that clarification. Let's see. Are there other questions here? Okay, if a patient does not qualify for medical cannabis, this is from Mitchell. Thanks for your contribution, Mitchell. If your patients do not qualify for medical cannabis via cannabis dispensary, what dose of CBD do you start patients on for pain conditions? And Mitchell also had a question about informed consent when you're prescribing or recommending cannabis and CBD. So please answer that question as well, because I think probably a lot of people have that question about how to handle the informed consent piece.
So for dose, I usually start people at 1 dropper full. With the admonition, they may need less, and just a few drops might do, but they may need more. And I have patients for insomnia or rapid cycling. They need something like 6 or 7 droppers full a day. But I start at one dropper full. Go ahead, Donna.
Well, I was going to say, and Dr. Carlton is talking about a 250 milligram dose per fluid ounce.
Yes, thank you. Thank you. And then I also, depending where the pain is, I tell them that they can apply it topically. And everybody says, topically? Well, it's important to know there are cannabinoid receptors in the skin. They're everywhere. The cannabinoid receptors are in every tissue of the body. So, yeah, they're in the skin. And my wife's aide, I was telling her about the CBD, and she told me that her mom has bad arthritis. So I said, here's a bottle of the Inner Health CBD. Tell your mom to apply it to her joints. So she gets the mom gets tremendous relief of her pain just by applying it topically. And neuropathy also can help topically.
Oh, interesting. I've not heard that before.
Yeah.
Now, Margaret is asking, is there any indication to use both full spectrum and isolate? Where you would use like a full spectrum plant extract and then also a CBD isolate.
I was just
go ahead. Please go. Yeah, please go, Luke.
Sorry about that, Donna. Don't mean to walk on you there. Yeah, there may be some rare instances where that could be helpful, perhaps where the patient just has a strong need for endocannabinoid stimulation at a high dose, and that dosage isn't cost effective with the whole plant extract. But even then, what we see with the isolate is that it has a bell-shaped response curve. With the animal studies on pain, and there's a human study on anxiety as well, that shows that at a high enough dose of CBD isolate, the effect is as if you're not taking any. And that happens because the CB receptor becomes overstimulated, and just like our insulin receptors, when people are overstimulated with insulin, it leads to receptor site desensitization. And we believe that happens because of the lack of terpenes. The terpenes have a resensitization effect because of their allosteric binding site. So definitely want that full spectrum effect. I'll see if I'll think about that any further, but I can't think of any other good applications for isolate. I think isolate is a cheap industrial product that is the commercial outlet for low quality hemp flower.
This is a little digression from that question, but it's something really important I want to mention. People think about the terpenes almost like an afterthought. Oh, and CBD. Well, the hemp plant and the cannabis and marijuana plant are so rich in terpenes, it's hard to put into words what the massive quantities of terpenes are compared to the most plants in the plant kingdom. It's just a super abundance of terpenes. And these have really potent roles, as some of my colleagues were telling me about before. And so a whole plant extract like EnerHealth, and if you see me plugging EnerHealth, yes, I am. I like to get a shout out and see people have a wonderful product. The whole plant extract gives you all these terpenes, myrcene and pinene, and it's a vast world of discovery going on to see what these things do. And you should know that there are companies in Colorado and California that are just extracting the terpenes and selling them onto the market because people want to take terpenes. So
interesting.
Yeah, in this product, you're getting a very rich abundance of terpenes because it's the whole plant and it's beautifully extracted at low heat, no pressure.
And there's a caveat to the terpenes being extracted. A lot of the terpenes that are being extracted right now are coming from other plant material. It's not coming from cannabis. It's coming from fruits, things like that.
Additionally, some of them are synthetic. But I had one more maybe note to add to that isolate thing. There, if you think about the evolution of the mammalian endocannabinoid system, there is no point in history where we have ingested, if you think about just the evolution of our system here, where we've been ingesting exogenous cannabinoids without the terpene content inherent in literally any plant that produces exocannabinoids. So it's just, our system is sort of designed for a whole plant or a whole food, I guess, therapeutic.
That's right.
Well, we have a couple of questions about the topical aspect. Somebody was asking whether the topical would be good for diabetic neuropathy. And,
yes, I believe it is. Yeah.
Okay. And somebody else was asking whether it's you have to factor in the potential cardiovascular contraindication when you're using topical formulations.
Oh, no. Topical, I think even if you use THC in a topical, you wouldn't have to worry about the cardiovascular complications because you're not going to get that much of any pharmacokinetically distributed through the body.
I think, your diffusion coefficients are going to be so low by the second membrane transfer that it would be, I agree with Dr. Carlton.
I have one patient on warfarin who cannot take cannabinoids orally for knee pain, but gets effective relief for his knee pain by using it topically, and there's no effect on the prothrombin time.
There you go.
The lowest therapeutic index drugs we can be pretty safely assured that topical is a very low level of interaction with the central nervous system and the central physiology.
Yeah, and the smooth muscle tissue.
Well, we're going to have to wrap up in just a few minutes, but I've been, there's sort of a big question I've been wondering for a while now. Given the high concentration of cannabinoid receptors in the gut, are there any interactions between the gut microbiome and the endocannabinoid system?
Great question. I don't know.
That's a fabulous question.
Yeah.
That may be some research that we need to do, Eric. I don't think there's anything on the literature yet that I've seen so far.
Okay, well, whoever gets the
I
Nobel
haven't seen
Prize
anything
first,
like
I hope you'll share it with me.
Yeah.
It's what I was thinking about it. You know, there's all these receptors, there's all these microorganisms, everything's in communication with each other. We know that the gut is so central to the immune system, and as they do more microbiome research, they're finding out how it affects things like sleep cycles and hormone release and all different kinds of interesting things on a micro to macro level. And so it just got me wondering whether the endocannabinoid system factors in there some way.
I have no literature that I've ever read on that.
That's a wonderful
apologize.
question.
Yeah, it's a
Okay.
beautiful question. No, I hope that that's where research goes next.
I think it's really important to keep in mind that CBD, as of only 10 years ago, was thought to be inert. I mean, you can find medical grade textbooks and the state of the science until really recently. We really just didn't understand that much about cannabinoid physiology. So we're in a very young area in medicine, and there's a lot more research to do.
Indeed. Okay, a couple more real quickies. This one I almost overlooked. It came in earlier from someone about the experience of getting sort of the GI system getting a little bit stopped up when they take CBD and having to kind of take THC again to THC to get things moving again. So an issue about gastrointestinal motility slowing down on CBD alone. Anybody insight on that?
Yeah, cannabis is the plant, has anticholinergic side effects. So, anticholinergic means you slow down motility and slow down the secretions. So that's a known factor in the pharmacology of cannabis. It's anticholinergic.
There is also, though, if you take the hemp seed, I know this is kind of mildly off topic. A lot of the fatty acid content in that has actually been used for thousands of years to deal with the constipation. So sometimes different parts of the plant help to make up for other parts of the plant.
Yeah.
I know that sounds like a ridiculous thing to say, but it's a traditional usage in a lot of places.
That's a great point, Donna. It's in Chinese medicine, and even after the communists came to power, they scrubbed away all of the psychoactive uses of cannabis from the Chinese pharmacopoeia. The one indication that remained was for the part of the plant known as huo ma ren, or fire hemp seed, which was used for, that's its main indication, was for constipation. My take on it is that people who are experiencing GI side effects probably have some other stuff going on with the gut, because in most patients with a fairly normal distribution of GI function, it should not be an issue because it's such a mild effect on the GI. It may be a dosing issue. The CBD and THC dosing may need to be experimented with to find a dosage of both that yields a good effect on the gut.
Great. Well, thank you all. I'm afraid I'm going to have to end this broadcast in just a minute or so. So if you do have other questions, feel free to reach out to the speakers. I know Dr. Carlton gave his email address. Steve, if you want to say your email address to answer any further questions that people have. Steve, you still with us?
Yes, I am.
What's the best email address for people who have questions?
Okay, Steve, STEVE at ENER, F as in Frank, OOD.com, enerfood.com. Conversely, they can send info at helixherbs.com.
And for my email address, since the slides were going up by pretty quickly, it's rcarltonmd, RCARLTONMD @gmail.com.
Very good. Well, thank you all for participating in this workshop tonight. I thought it was really fascinating. Of course, generates as many questions as it. That's the process of science and the process of progress. So stay tuned. I'm sure we'll be hearing more from the Helix Institute for Health. I know they've got some other educational activities planned in the future. So stay tuned for that, and so for some other webinars coming up from Holistic Primary Care. We do appreciate your participation, and we hope you found this helpful, because obviously we want to provide you with information that can help you do the best possible job helping your patients. So with that, I'm going to sign off. My name is Eric Goldman. I'm the editor of Holistic Primary Care. If you have questions about Holistic Primary Care or just want to get in touch with me, I'm at Eric, E-R-I-K, at holisticprimarycare.net. So have a wonderful night, and we'll connect with you again very soon.
Thank you, Eric.
Thank you.
Thank you.
Thanks a lot.
Great.